Good morning, students. Let us continue with the next organelle, plastids. Plastids are found in all the plant cells and in euglenoids. Remember euglenoids, member of kingdom Protista? These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on this type of pigments, plastids can be classified into chloroplast, chromoplast and leucoplast. Remember one more thing, that plastids are found in plants only. The chloroplasts that contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophyll and others are present. This gives the part of a plant a yellow, orange or red color. The leucoplasts are colorless plasmids of varied shapes and size with stored nutrients. Amyloplasts store carbohydrates, that is starch. Example, potato. Elaioplasts store oils and fats, whereas the aleuroplasts store proteins. Majority of the chloroplasts of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid or even ribbon-like organelles having variable length 5 to 10 micrometers and width 2 to 4 micrometers. Their number varies from 1 per cell in Chlamydomonas, a green alga, 20 to 40 per cell in their mesophills. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. Permeable means it allows only few solutes to move across. The space is limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stacks like the piles of the coin called grana. Each is known as granum or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids enclose a space known as lumen. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigment are present in thylakoids. The ribosomes of the chloroplast are similar, that is 70S as the prokaryotic and cytoplasmic ribosomes as 80S. Here is the structure of chloroplast. We can see the double membranes, outer membrane, inner membrane. Granum, intergranal lamellae or stroma lamellae, thylakoids. Each these coin-like structures are there. Each coin is thylakoid and the fluid present in the lumen of the chloroplast is known as stroma, which is rich in several enzymes and ribosomes as well as small circular DNA. Next organelle is ribosome. It is a granular structure first observed in the electron microscope as a dense particle by George Palade in 1953. Underline the name of the scientist who observed it first. They are composed of ribonucleic acid, that is RNAs, and proteins. These are not surrounded by any membrane. So these are just the particles made up of rRNA, that is ribosomal RNA and protein. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller. The two subunits of 80S are 60 and 40 while of 70S are 50 and 30. Listen, how to remember these units. Every time you have to minus 20. If ribosome is 80S, then 80 minus 20, 60, 60 minus 20, 40. So 80 के सब यूनिट्स हो गए 60 और 40. वैसे ही 70 में से minus 20, 70 minus 20, 50 and 50 minus 20, 30. This way you will get the units, because if you add up the units, it will be more than the actual number. So don't add it, just keep the rule in your mind that you have to continuously subtract 20 from one another. Here, S, that is Swedberg's unit, stands for the sedimentation coefficient. It is indirectly a measure of density and size of the ribosome particles or ribosome units. Both 70S and 80S ribosomes are composed of two subunits. We have seen that 70S are found in prokaryotes, mitochondria and plastids, while 80S ribosomes are found in cytoplasm of eukaryotes. Here is the structure of ribosome representing its larger unit, smaller unit, both combine to form a structure known as ribosome. If it is 80S, then the larger unit is 60, small is 40. If it is 70S, the larger is 50 and smaller is 30. So this way, these are the ribosomal particles which do not have any membrane. They are just made up of RNA and protein. Next part of the cell is cytoskeleton, an elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments and intermediate filaments present in the cytoplasm, collectively referred to as cytoskeleton. Three things are there: microtubules, microfilament, intermediate filament. In a cell, it is involved in many functions such as mechanical support, motility and maintenance of the shape of the cell. Next is cilia and flagella. Cilia, singular cilium, flagella, singular flagellum, are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars and causes the movement of the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement only. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. The electron microscopic study of cilium or flagellum show that they are covered with the plasma membrane. Their core is exoneme, which possess a number of microtubules running parallel to the longitudinal axis. The exoneme usually has 9 doublets, means 9 pairs of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of exonemal microtubules is referred to as 9+2 array. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublet by a filament known as radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are interconnected by linkers. Both emerge from centriole-like structure called basal bodies. Here is the diagrammatic representation and electron micrograph of the cilia or flagella. We can see in the diagram how the plasma membrane is covering all the doublets. There are 9 pairs, that is 9 doublets of the peripheral microtubules. At the center are central microtubule. Red colored sheath is present, that is central sheath, and radial spokes are there. All the doublets are connected by the interbridges. Centrosome and centrioles. Centrosome is an organelle usually containing 2 cylindrical structures, centriole, which are perpendicular to each other. They are surrounded by an amorphous pericentriolar material. So, these have an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each peripheral fibril is a triplet. जैसे पहले 2 की पेयर थी, यहाँ पर 3 की पेयर्स हैं. Adjacent triplets are also linked. पास वाले triplets भी linked हैं, और central part of the proximal region of the centriole is also proteinaceous and called hub. वहाँ पर exoneme कहा था, यहाँ पर hub कहेंगे. It is connected with the tubules of the peripheral triplets by radial spokes made up of protein. The centrioles from the basal body of the cilia or flagella and spindle fibers that give rise to spindle apparatus, that is helping during the सेल डिविजन इन द एनिमल सेल्स. सो सेंट्रियोल के 3 मेन फंक्शंस हैं. पहला सीलिया फ्लैजिला की बेसल बॉडी बनाना, दूसरा स्पिंडल फाइबर्स बनाना जो कि सेल डिवीजन में मदद करते हैं. सो द नेक्स्ट, लास्ट but not least, the main organelle of the cell, the control center of the cell, or we can say the brain of the cell, is nucleus. It was first described by Robert Brown as early as 1831. Later, the material of nucleus stained by the basic dyes was given the name chromatin by Fleming. The interphase nucleus, that is nucleus of a cell, when it is not dividing, it is at rest. It is known as interphase nucleus. It has highly extended and elaborate nucleoprotein fiber called chromatin. जब सेल डिवाइड नहीं हो रहा होता है उसके अंदर सारा जेनेटिक मटेरियल इन क्रोमैटिन के फॉर्म में रहता है. न्यूक्लियर मैट्रिक्स एंड वन और मोर spherical bodies known as nucleoli. Electron microscopy has revealed that nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer, is called perinuclear space, forms the barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and it bears ribosomes on it. At a number of places, the outer membrane is missing or it is interrupted by minute pores which are formed by the fusion of these two membranes. These nuclear pores are the passages through which the movement of RNA and protein molecules takes place in both the direction between nucleus and cytoplasm, that is in and out. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. We must try to know the names of the organisms that have more than one nucleus per cell. Some mature cells even lack nucleus. For example, mammalian RBCs, that is erythrocytes, and sieve tube cells of the vascular plants. Will we consider these cells as living? Yes, of course, because all the important metabolic reactions are taking place inside these cells without nucleus but are governed by the nucleus of the nearby cells. The nuclear matrix or the nucleoplasm contain nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. जैसे सेल के अंदर साइटोप्लाज्म वैसे न्यूक्लियस के अंदर nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not membrane bound structure. It is site for active ribosomal RNA synthesis. Large and more numerous nucleoli are present in the cells actively carrying out the protein synthesis. Here we can see the structure of nucleus. It is an electron micrograph of a nucleus in which we can see the nucleoplasm, nuclear pores, nuclear membrane and the dark spherical body nucleolus. We may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fiber called chromatin. But during different stages of cell division, cells show structured chromosomes in place of nucleus. Chromatin contain DNA and some basic protein called histone and some non-histone protein and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 chromosomes. You will study the details of the DNA packaging in the form of chromosome in class 12. Every chromosome that is visible only in the dividing cells. Non-dividing cells have chromatin, dividing cells have chromosomes. Essentially has a primary construction called the centromere, on the sides of which disc-shaped structures called kinetochores are present. मतलब क्रोमोसोम जो है बीच में से सिकुड़ा हुआ है और उस सिकुड़े हुए स्ट्रक्चर को सेंट्रोमियर कहते हैं, जिसके दोनों तरफ प्लेट लाइक डिस्क लाइक स्ट्रक्चर्स हैं जिन्हें कहते हैं kinetochores. Let me show you the chromosome. Here is the chromosome fragments जो दोनों तरफ से बीच में से सिकुड़ा हुआ है और फिर ग्रीन कलर की डिस्क दिखाई गई है, दैट इज kinetochore. Centromere holds two chromatids of a chromosome. Based on the position of the centromere, chromosomes can be classified into four types. The metacentric chromosome has the middle centromere, forming two equal arms of the chromosome, while the submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas telocentric chromosome has the terminal centromere. So, absence of two arms, only two long arms are there. Let me show you four types of chromosomes. We can see the position of the centromere different in the different chromosomes. First one is metacentric chromosome, center centromere, equal arms. Second is submetacentric, one longer, one shorter arm. Third is acrocentric, one very long and one very short arms. And the last one is telocentric, in which the shorter arm is almost negligible. There are some constrictions, secondary constrictions, that forms satellites. Sometimes a few chromosomes have non-staining secondary constrictions at a constant location. This gives appearance of small fragment known as satellite. Such chromosomes are known as satellite chromosomes. Next is microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. This was all about chapter number 8.